Hello and welcome to the 11th video for our course in Diversity and Evolution of Insects. We've been spending quite a bit of time talking about how one comes up with a phylogenetic hypothesis of relationships amongst organisms, the different methods, the different approaches, and the philosophies behind them. But we really haven't talked about how you go from a hypothesis of a phylogeny of a group of organisms to actually do something with it. And so what I want to focus on for this video are two areas in which you actually use a phylogeny. The first is to produce a formal classification from the phylogeny. And the second is to get an idea of when in the history of the planet certain events took place. When did the splits? When did the dinosaurs arise? When did the birds arise from the dinosaurs? When did winged insects first arise? We want to get a handle on the attempts to put absolute ages on some of these evolutionary events that are reflected in our phylogeny. There we go. So let's start with classifications. I think I probably have been in the words I have used have kind of equated at times the phylogeny with a classification. And in one sense, that's, you can understand, I hope you can understand the natural inclination, because both a phylogeny and a classification are hierarchical in structure. And one, the classification, should be derived from the other, the phylogeny. So sometimes probably the verbiage I've used has been a little bit ambiguous because in my mind I was subconsciously equating some of these things. But they are two different things. A classification, the formal Linnaean classification, there are sets of rules that are involved in classifications. Most of these get to how do we name the taxa within a classification, and I'm going to put a hold on the rules of nomenclature for the time being. We'll come to that soon enough, and rather talk about given a phylogenetic hypothesis, this hierarchy that we've created these days using phylogenetic, that is cladistic techniques. How do we translate that into a hierarchical classification? Well, we have a good start because we have a hierarchy in both cases, and we don't want our classifications to be to contradict the phylogenetic hypothesis. But then the question becomes just what elements in the phylogenetic hierarchy that we've come up with from our massive molecular data, for example, which elements of that do we want to be reflected in the classification? And this kind of begs the question, what the heck is a higher taxon anyway? Where are those things defined? What was it, the second class that we spent a whole video, and I probably rambled on for well over an hour about, what is a species? How do we define a species? And that was only one level in the entire Linnaean hierarchy, where people have argued over and pondered over and come up with dozens and dozens of different ways to define species. Is the same thing going to be true at these other levels as well? Are you going to have to put up with me talking about what a genus is for more than an hour? Well, you're probably quite happy to learn that the answer to that question is no. While there's a lot of discussion and hypotheses and back and forth about what a species is, there are frankly painfully few guidelines about what the other ranks in a classification are. In fact, if you go to what definitions do exist are very simple and simple-minded. Well, I'll add some constraints here in a little bit on these, but basically a genus is nothing more than a group of species. There are no established rules about how to determine which species can be put together in the same genus and which can't. There's lots of leeway there. And so that's well, there's the facetious sort of definition of what a genus is that I have heard some taxonomists claim, and I believe Willis actually referred to this in that reading you had, and that a genus is whatever a good taxonomist says it is, which is not very helpful, and certainly not helpful for somebody who's using the classification to be able to understand the underlying basis of it in facts. But putting facetiousness aside, a genus is little more than a group of species. A family is basically a group of genera that a classifier, a taxonomist, has put together into that same taxon. An order is nothing more than a group of families. So by putting it that way, I am implying in some ways that there are absolutely no limits on what species can go into forming a genus, but that's not quite true. So let's examine this a little bit more closely. So if we're going to group species together into a genus, what's the basis that we're going to be doing that? Some people, of course, have advocated even within a phylogenetic context, in the context of having a cladogram at hand for the group in question, that the similarity should play a role in our choices of classification. Of course, this harkens back to the days of phonetics and the days of evolutionary taxonomists, which thankfully are not entirely gone, but that are definitely not the predominant mode by which classifications are made anymore. But even if you are working within a phylogenetic context, the idea that we want the organisms within our higher taxon to all be very similar to one another runs into some real problems. First of all, can similarity be objectively measured? Or maybe the way to really say that, since we've talked about ways, a simple matching coefficient is one measure of similarity. The real problem is there a common definition of similarity upon which we could all agree? And I suspect the answer to that is no. And I believe that one of the reasons that phonetics faltered as a dominant philosophy for taxonomy was not only that it ignored evolution, but that they were able to propose literally dozens and dozens of different methods, all of which are mathematically sound, all of which do the task for which they're defined, but there's no real way to choose amongst all of them because they don't all give the same answer. So if they're going to differ, which one are we going to accept? So can we objectively measure similarity, and can we decide upon a common measure of that similarity? But something even more troubling is that we have to ask ourselves the question of whether or not a particular characterization of similarity can be exported from the context of talking about insects, such as the two rediviid bugs I have there on the right-hand side, to talking about organisms in a totally different group, like the two mice on the left-hand side. There's also an interesting corollary that it seems that the more well-known a group of organisms is, the more they get subdivided, because if you know something really well, you are able to spot the differences between them that make them dissimilar in your eyes as one of the cognoscenti, as opposed to somebody who just stumbles in and is looking at these things for the first time. For example, the two insect pictures there. I see vast differences between these organisms. I also understand what the similarities are, but if I brought my granddaughter in and said, look at these two things, are they the same or different? She'd probably say, well, they're the same because they're two bugs, right? And in this case, they actually are bugs. So another manifestation of that is that while the two, to me, quite different looking creatures here on the right-hand side, the the ambush bug up here and the thread-legged bug down here, they look so very different. These are classified in the same family in, I'm sure there's an exception, but in most classifications of true bugs. Whereas these two mice, which to me, for all the world, look about as similar to each other as anything could, they're actually classified in two different families. The murids and what are these? The Paramiscus chrysetids, the family here. So the more well-known a taxon, the species of a taxon are, like these mice, the more we have this human tendency to subdivide them because we know them so well, and we can understand the minor little differences that distinguish them. We ascribe great importance to that. That's just something about the human psychology that I think is always going to be part of the equation that we have to keep in mind. So if we can't agree on a measure of similarity that we're going to hang our hat on when we make our classification, are there other options? Well, yes, the one other option that has been proposed, and by no less a luminary than Willy Henning himself, was that we could decide where to draw the line on which species go together in a genus and which genera go together in a family based upon the age of the organisms, how old these groups are. When did they first arise in the fossil record, in the history of the planet? So actually, I think I'll have it on the next slide. I think age has been proposed to be an objective measure by which we could say that families of rodents and families of Hemiptera are similar in that they both arose at roughly the same time, if we were using age as the criterion there. Issues with this, of course, are that how do we know how old a group is? And that's going to get into the second topic of this video. But traditionally, especially in the context of when we only had basically morphological characters by which to group organisms and try to figure out their phylogeny and evolutionary history, then we could use fossils. But not all groups have fossils, and not all of the nodes in a cladogram will have fossils that can be assigned to that node. So that becomes a bit of an issue. Let's take a look at this next slide here. Yeah, this is an image that was taken from, actually it's redrawn from, but it's basically one of Hennig's images that he published a book called how do you translate it? The Phylogenetic History of Insects, in which he applied his cladistic techniques to look at the overall history of insects and how they should be classified. So what we have here is, you can recognize, I think, the cladogram, the underlying cladogram in this part of the tree, and these are the names of taxa. I'm not going to focus on them for the time being. The vertical axis here is the time scale. So we have Cenozoic, Cretaceous, Jurassic, Triassic, Permian, Carboniferous, and the Devonian eras. And so we see some very early fossil insects / hexapods, and we'll get into this when we talk about the wingless hexapods, appear very early in Devonian era. But let's say, just for the sake of argument, that we're going to draw a line here at the division between the Triassic and the Permian, and that any group that is older than that timeline, dividing those two geological periods, any group that arose before that, we're going to classify as orders. And so what do we have here? We have mayflies appearing here. We have, that's going to be the dragonflies, the Odonata, and then all the other ones that are appearing along the way. Anything that appeared later, and in this particular illustration, he's focusing on the evolution of orders, so we're not getting a lot of branching, explicit branching up here, although you can see in the width of the line, he's trying to give an indication of the diversity, the number of species that occur within each of these groups. But he was drawing a line here between the Triassic and Permian and saying anything that arose before that classify as a separate order. So that didn't really work too well, because it's very difficult, as I hope I'm going to make clear to you shortly. It's pretty difficult to assign absolute ages to taxa, and there's a lot of room for disagreement, differences of opinion. And of course, when we have a classification, a formal classification, because we want this to be used by non-taxonomists, we want this to be the general reference system by which we organize everything we know, physiology, behavior, molecular biology, everything we know about organisms within that classification. We really don't want a system that's going to be changing every other year or every 6 months, or these days with electronic publication every other week or something like that. So, I lost my train of thought there for a second. What rules can we apply here to kind of circumscribe the entire universe of options for classifying things and having disagreements and fighting systems, as you will? Can we reduce that? Well, there are a few rules of thumb that I guess I'll make the last point on this slide first. These are rules of thumb, but there are absolutely no ironclad rules that prescribe or prescribe how to define and create higher taxa by taxonomists. The only thing that is a constraining force external to the systematist himself is the peer pressure of the people who are going to be reviewing the publication. And if the publication isn't peer reviewed, then there's really nothing there at all, probably. That is what non-taxonomists, non-systematists, when I talk to them about this kind of stuff, they point out and they just scratch their heads and say, "What are you guys doing?" We're doing a hell of a job, but let's start right there. And there are guidelines that prescribe how to go about this. There are always going to be wildcats. What would we call them? People that are going their own way, and it's kind of going to end up being simply the consensus of opinion that relegates those sorts of classificatory schemes to the dustbin of history eventually. Knock on wood, hope, cross your fingers, hope to die. Anyway, the rules of thumb. First of all, I want to bring up the issue of what are called monobasic taxa. All this means is that you have, let's say, a family, and if that family contains only a single genus, then we would say that the family is monobasic, or sometimes you'll hear it called monotypic. So what's wrong with monobasic taxa? The classification is replete with monobasic taxa. Some of these things arise because some groups are just so old that if there were other species or genera, depending on the level we're talking about in the classification, that would be classified with them, they've gone extinct. For example, the tuatara off of New Zealand. There's nothing close to those creatures, and so there's basically only, I think there's 2 species and 1 genus of tuatara in a family, in a superfamily. So we have a whole series of monobasic taxa there. In other cases, monobasic taxa are a reflection of our ignorance, that we've discovered some crazy weird insect. Let's say we were collecting in southern Chile, down there, former area of Gondwana land, and we find these weird creatures. All one species, they don't fit in any other genus. We need to put them in their own genus. And so now we have a monobasic genus. But history has shown us that very often we end up discovering more species in this example that belong to that genus, and so it's no longer monobasic. And so maybe you could think of it as either an expression of ignorance because we haven't collected the other things, or as a placeholder for the time when we do discover more species that belong in that genus. So the fact that there are so many of them, and we can understand how they work and why they might deserve to be in a classification. What's the problem? Well, the problem is that if my premise that our classification is designed to be the overall reference system by which we store information, then having a family that contains only one genus, that family serves no function. I mean, it doesn't, the information storage capacity of a taxon is already being fulfilled by that genus. And having a single family for that, single genus is simply redundant. We gain nothing in the process. So by and large, I think the rule of thumb is, at least in my own philosophy, is to try to minimize the number of monobasic taxa you're dealing with, because they don't really add anything to the equation. The second point, though, is something that I think I think is widely adhered to these days. And if you tried to propose a reclassification in any reasonable professional scientific journal these days, if the taxa you're proposing are not strictly monophyletic, you are going to get a lot of pushback from peer reviewers, and quite possibly a lot of rejection at the same time. So when people propose higher taxa these days, anything from genus up to order, these taxa must be, as far as I can push that phrase, strictly monophyletic. And I mean monophyletic in the cladistic sense. That is the common ancestor and all of its descendants. So no paraphyletic groups, no polyphyletic groups are acceptable. That's the gold standard anyway. The third point, the Hippocratic Oath, what do I mean by that? Basically, all I'm saying there is the first rule should be to do no harm. And by harm, I mean coming into an established, if perhaps not ideal, classification, and just turning everything upside down because you have the power to do it, to come in and reclassify everything so that all of the terminology and the understanding that people had about how organisms were grouped together, and therefore they were able to store that information about the groups by using those names, to come in and turn that upside down simply because you can, I think is bad form. So, I mean, of those three constraints that I've placed there, the only one that's really got any teeth in terms of the general community that you can expect the general community to try to enforce is the constraint of monophyly. The other two, I guess, are more my own preferences, and I think good practice, good professional practice, what do they call it, best practices, than anything that's written in stone. And just to reiterate that last point, there is no governing authority for this. In the long run, it is the consensus of scientific opinion. That is, which classifications do people end up using? And it's not unusual that we have multiple classifications out there that are coexisting more or less peacefully, sometimes not so peacefully, at the same time, and thereby, frankly, generating a lot of confusion. So let me give you an explicit example. This is from the research that we do in the lab. So I think I started out this course telling you that I we work on parasitoid wasps in the order Hymenoptera, and with our focus on the superfamily Platygastroidea, most of which are egg parasitoids of other insects. Actually, let me get into that a little bit. Classically, the superfamily Platygastroidea, and when I say classically, I'm talking from about 1930 or so up until 2007. So all of the species in this superfamily, and there's now, the last count I have is something like 6,000 species in this superfamily and growing every year. They were placed into two families. So there was the Platygastridae and the Celeonidae. The Celeonidae, as far as we know, are all egg parasitoids of other insects or spiders. So they have a common biology. The Platygastridae, on the other hand, there's about 1,100 species of platygastrids. The vast majority of those attack either the eggs or the larvae of gall flies, the family Cecidomyidae. And so, you know the size of gall flies. These are minute little flies, and so they're attacking the eggs or the larvae. So these are minuter little wasps. But this division into two families roughly corresponded to the biological difference. Okay, in the platygastrids, there are a few egg parasites too, and there's some that do weird stuff. You don't really understand. But basically, we have these two families, and it's kind of biologically based. So that was the intuitive classification. There had never, up until Murphy et al. 2007, which is where I'm going next, there had never been a comprehensive analysis, quantitative analysis of relationships on any source of data, morphological, molecular, whatever, for this group. That changed when Nick Murphy, who was a postdoc in the University of Adelaide in South Australia, working in Andy Austin's lab. Together, they and a few other co-authors published the first phylogenetic analysis for platygastroid based upon, 3 molecular markers. If I can get my cursor there, I'll go to it. So here are the results. And like any phylogenetic tree, the first caveat is, I know you can't read that, so don't worry about it. Anyway, what we have here is the overall tree for the phylogeny. And let's see, the outgroups have been pruned out. The numbers, this is a Bayesian tree, so the numbers represent the posterior probabilities of the of these groups being true. Let's see here. So we classically had two families, the Platygastridae and the Celeonidae. And up here in this top red box, we see that it contains, and if you can't tell, here's the branch leading to all of the OTUs that they had on their study for the that they had in their study that belonged to the Platygastridae. They all grouped together in one group. Everything else, classically, was a Celeonid. The other two boxes refer to subfamilies. We don't need to focus on them at the moment because I want to restrict our attention to the family classification. So, platygastrids, perfectly monophyletic group. We're good there. They could be a family. But what about the rest of the species within the Platygastroidea that had always been classified as Celeonids? Well, we see a big chunk of them down here. The vast majority, as a matter of fact, arising here. I want to point out this node right here that has 100% posterior probability. Every analysis we do with molecular data, with 3 genes, 4 genes, 4,000 genes, that node always shows up. That's about as true as you can get. However, we see a branch here above this, what they call the main Celeonid clade, leading to Celeomorpha and Sparazion there. And then at the base of the tree, we see another couple of taxa, Archaeotelia and Neurocelioidae there. What does this tell you? This tells us that if we recognize the platygastrids as a taxon because it is a monophyletic group, that renders the rest of these species, or to put it more precisely, that renders the classical family Celyonidae as a paraphyletic group, because here's where the common ancestor would be. It includes a good deal of, but not all, of the descendants of that common ancestor. So we end up with one monophyletic group, one paraphyletic group, which is the middle no-no that I pointed to in the constraints on how we're going to come up with a family classification. We can't recognize a family Celyonidae because it's paraphyletic. It's not monophyletic. So that result led a colleague, Mike Sharkey, down at the University of Kentucky, to look at the results of Murphy et al. Murphy et al. Were kind of, okay, this has got to be looked at as a bit preliminary because the number of species we have in our analysis is only a minor subset of all of the taxa that have been classified here. So we need to be a little bit careful. Well, having published the result, that did not constrain anybody else to be as conservative as, or as careful. And Mike said, well, you know, if Celyonidae is a paraphyletic group, then we can't recognize Celyonidae in our classification. That leaves us with only really one solid conclusion that is supported by the data, and that is to say that we will only recognize a single family within the superfamily Platygastroidea. And because of the rules that we're going to get into soon enough, its name should be Platygastroideae. So suddenly we go from a superfamily with two families to a monobasic superfamily, a group that contains only one family. And, you know, as I said, what does that do in terms of providing structure in order to organize your knowledge? It does nothing. It's totally redundant. Some authors, Mike Engel and Ryan McKellar, for example, noted that, boy, if you do that, if you just collapse this classification into a single monobasic superfamily, then you're losing a lot of information about the biology, the biological difference that was inherent between where are they Platygastroideae, most Platygastroideae, and Celyonidae. And you're losing lots of information about the subfamilies within there, those other red boxes I was showing. And so they said, no, the Sharkey classification is no good. We should recognize not one, and we can't recognize two because we agree that a paraphyletic Celyonidae is not a good thing. We think there should be 4 families there. And so they said we should recognize the traditional Platygastroideae, that main Celyonid clade as the newly circumscribed family Celyonidae, and then 2 other families, the Nixoniidae and the Sparazionidae. Let me show you where those guys come into the equation. So here's the Murphy et al. Cladogram again. And so McKellar and Engel are saying, okay, here's the main Celyonid clade. All these guys from here to the bottom, Celyonids. All these guys up here in the red box, Platygastroids. Celeomorpha and Sparazion here, we should recognize those as the family Sparazionidae. Ooh, there's no, I said Nixoniidae, another family. Actually, the analysis of Murphy et al. Did not have any species from that. There's only a single genus there, Nixonia. Did not have any species of Nixonia in the analysis. So Engel and McKellar based that upon, I don't know what they based it upon because they didn't tell us what they based it upon. And it also leaves hanging the question of, well, okay, we dealt with this, we dealt with these 2 guys here, 3, Nixonia is outside. What about these 2 up here? Archaeotelia and Neurocelio. In fact, Archaeotelia was traditionally classified together with these guys here, but now it's broken up off at the top. No family proposed for this. Neurocelio, they didn't even mention. So this kind of stuff was just left hanging. Now combine that with the fact that between 2007 and the present day, in a lot of the research we've been doing, we've been discovering lots of new, really strange species that don't fit into this classification at all. We don't even know, we can't even say where they fit anywhere, except that they belong to the superfamily as a whole. And so a new classification is certainly needed. We've got these competing classifications. We've got Sharkey's single family, we've got McKellar and Engel's 4 families, and of course a lot of people still cling to the traditional 2 family use. So it's just been total confusion. So one of our objectives over these all too many years is to develop a data set and analysis, come up with phylogeny for Platygastroidea, and translate that into a robust, stable classification for the entire superfamily. So let me show you where we are and the kind of decisions we're going to have to be making. So here you see it's actually a single phylogenetic tree that I've broken into 2 parts. So if we look at the one on the left, this is the base of the tree. Actually, these up here, those 4 at the top are our outgroups. And so the tree kind of goes down from here. Ignore the color for the time being. And this box shows you where in the overall phylogeny this branch, this part of it, belongs. And then you see the down arrow here, that continues here with all of these guys here, and see the second half. So actually you would want to glue this to the bottom of that to see the classification. So we've got tons of taxa here, well over 100. We've increased the representation of genera to a very reasonable number, but obviously we're not going to do all 6,000 species. It's both practically impossible. Well, it's practically impossible because you can't get material from any species that's in good enough condition or to extract DNA. And it's not necessary. We're assuming at some level that these OTUs represent monophyletic groups. That's generally a good assumption. There are some genera here that probably don't warrant that, but we're actually grouping together species here because this is all based upon, well, actually it's based upon a combination of molecular morphological data. So Murphy et al. Had 3 genes. We upped that to 4 to include a nuclear protein coding gene and added a ton of morphology. Even with doing so, the real question, this blue thing here, corresponds to the main cellionid clade. So it's up here where we want to focus our attention. And you can't see it, but these numbers, these are bootstrap numbers, because this is a maximum likelihood analysis. These are not encouragingly large, shall I say. That is the confidence that I feel that we can place that these nodes are correct is not high enough to warrant building a classification on them because we think they're too liable to change. So we decided to supplement this data by, instead of just doing it 4 molecular markers, then morphology actually just confused the situation more than anything else, to be honest with you. Let's expand this in a phylogenomic approach using that ATRAM protocol to 4,000 genes. Now to do that, we couldn't do that on all of these OTUs, and so we only did it on a subset. This whole blue part here, we only have represented by 4 species, a little bit more up in the more basal part of the tree though. So here's the results of that analysis. So now we flipped it on its head. This is that blue part of the tree that I had up before with the 4 representatives. And just for orientation, these genera belong to each of the 4 subfamilies that had traditionally been recognized in the in the Celyonidae. These 3 are outgroups, and so here's the core of our question here. How are these guys interrelated? Now you notice there are no numbers. to give you any branch support indication there. And that's because the bootstrap support value for these is all 100% for the genes. Let's see, what else do I want to point out? We have a Nixonia here in our analysis, and so that's the Engel and McKellar family. We also have the Neurocelio that was at the base of the tree in Murphy et al. Whereas Archaeotelia is the other one that was at the very base of the tree. So to walk you through this clade here from this node, Trichosis, Amitus, Zelostemma, and Inostemma, those are all classical platygastrids. And so my preference would be to recognize those as a family. Remember that the constraint here is all the families I recognize, I want them to be strictly monophyletic. So that we want to recognize that as a family. Here's the main Celionid clade that in all molecular analyses comes out with 100% branch support. I'm tempted to recognize that as a family. So then Engel and McKellar, they recognize Platygastridae, Celionidae. Then they recognize a thing called Sparazionidae. There's the name, the genus after which that family is named, Sparazion. That groups together in a monophyletic group that now includes Archaeotelia, where they had been traditionally classified. So that looks like it could be a good family. But what to do down here? Well, Neurocelio plus Nixonia forms a monophyletic group. So, strictly speaking, I could follow the rules and recognize that as a family. But here's where the do no harm mandate comes into effect, is that Nixonia is a big whopping wasp. It's a centimeter long, some of them. They're quite large, whereas Neurocelio are little bitty guys. And to be honest with you, I cannot find any characteristics that if I gave specimens to you to look at and key out under the microscope, that I could say I can't find any characteristics that unite Neurocelio and Nixonia. Apparently there are some molecular characters, but morphological characters, I can't find them. So to base a family, to describe a family, or to accept a family that I can't tell you how to recognize, seems like craziness to me. We've decided that, okay, we will accede to McKellar and Engel's separation of Nixonia off as its own family, a monobasic family. Don't want to do it, would prefer not to, but I don't see any better solution, which then also leaves us with another monobasic family here, Neurocelio. So there's actually two other taxa that we couldn't do phylogenomic work with that also fall into this area of the base of the tree that we're going to recognize as separate families. Yet another one that's monobasic, but the last one has two living genera and at least 2 fossil genera. So it's got 4 genera classified in it. So it's not monobasic. So instead of the 1 family classification of Sharkey, the 2 family classification, which even in this phylogenomic analysis renders Celionidae paraphyletic, or I guess our new classification is consistent with the 4 family classification, although at least we define how we got to our conclusions. But then that's 4, then we're adding 1, 2, 3 more for 7 living families, and then there's yet another fossil family that of course we couldn't do the molecules on. So we're going from the current 1 or 2 or maybe 4 family scheme to 8 families. Now of course that's not the entire gamut of classifications that I could legitimately come up with and defend as all of them being monophyletic. Even if, of course, the trivial solution was recognize everything as its own family. Do no harm again, the Hippocratic Oath. That would be that way would lie madness. We could, for example, have recognized this node as the new cellionity, which would recreate a lot of the old cellionity with the exception of these guys down here. Or we could separate Archaeotelia off as its own family. It's a weird thing. It certainly would deserve it on the count of similarity. But I can tell you how to identify a family sporazionity that includes all of these groups. So there is no magic bullet here. There's a level of art, if you will, to know where the sweet spot is in a classification. And personally, I think that comes from the people who have a detailed familiarity and understanding of the characteristics of these organisms in order to come up with the best way. There's not an algorithm that would be applicable here and then would be applicable to flies or grasshoppers or even those darn little mice. So that's my real world example. We're going to submit this paper for publication probably this week. There's the layout of the family with the colors back now. So now you know what the colors in the previous slide actually meant. Okay, so let's go back to talking about the process of dating. It is a natural, well, if you look at a cladogram, it's important to understand that what a cladogram is a representation of the hierarchical distribution of derived character states among the OTUs in the analysis. I know that's a mouthful, but if you think about it, we're just talking about how the characters are distributed and we're inferring from that what that tells us about relationships among them, and implicitly something about the timing of the speciation events that gave rise to the diversity we see. But the time dimension is not explicitly incorporated into a cladogram. Strictly speaking, a cladogram shows you the distribution of character states. But it doesn't take much of a leap to come to the conclusion that the cladogram does imply something to you concerning the temporal sequence, the sequence through time of the splits that led to this distribution of character states. So there's some concept of relative time there in a cladogram, in a phylogenetic tree, but the absolute values are not there. So I think it's a natural human desire to put numbers on this. When did angiosperms evolve? When did green plants evolve? When did insects evolve? We would like to know when, or at least it's an open question, and all open questions beg for answers. So classically, the way that this has been done is to hope that you find a fossil that you can assign to the taxon that you want to date. And then you date that fossil by physical-chemical techniques. Put an age on that fossil. And what that tells you, what that would tell you is what the minimum age of the taxon is. The taxon might be older because, is this an early fossil, a late fossil? This group could have evolved earlier, but you didn't get a fossil until a little bit later. So it tells you the minimal age of the taxon. And so for decades, centuries really, this was the way that we put hard numbers on how old various groups were. And this method has a number of issues that I'm sure will leap to your mind. First of all, there are many groups for which there is absolutely no fossil record. The fossil record is so sparse that there's no way to do this. Secondly, it may be difficult, even if you have a fossil, it may be difficult to know where on your phylogenetic tree that you developed on the basis of the study of modern extant organisms, where that fossil really should fit there. And so placing fossils can be an issue. And then finally, we saw in our molecular tree that there can be uncertainty in the topology of the tree. And if you're basing your timing of clades on putting fossils within that tree, then the uncertainty in the tree is going to translate into uncertainty in the ages of the taxa as well. All of which are problems with this. So, how can we address this? Here's where the idea of molecular clocks comes in. Actually, molecular clocks were first proposed in the early 1960s. So you remember Watson and Crick, structure of DNA, the idea of the sequence of bases in a DNA molecule, encoding genetic information. So when was that? That was the early 1950s. So about 10 years later, Emil Zuckerkandl and Linus Pauling, I might have spoken about Linus Pauling before, actually had the insight that certain biomolecules carry information about the history of the organisms that possess them. And they pointed specifically to DNA, RNA, and proteins, that these contain information. And so this was about the time, that you could do sequencing of proteins and nucleic acids, but it was painstakingly slow, expensive, and difficult to do. It's nothing at all like what we have today. But Zuckerkandl and Pauling had the insight to see that if you're looking at orthologous proteins, and then you're comparing the same sites in these orthologous proteins. So to translate that into modern terms, we're talking about a multi-species alignment for orthologous proteins. Then you can compare and basically count up the number of differences between a pair of organisms. And that accumulation of differences has arisen from the net impact of mutations that have taken place between now and the time when those two organisms shared a common ancestor. And so those mutations have accumulated over time. The number of mutations is proportional to the time that they have been from the time that they separated from their most recent common ancestor. And so now, if you can, in some way, maybe through fossils or some other way, know when that split, when those two species, let's put it this way, when those two species that you've been comparing in your alignment of their proteins, when they had the most recent common ancestor, then you can calibrate the rate of mutation in that protein. And by doing so, then by simply looking at the number of differences in the sequence between a pair of organisms, applying that rate that you know that protein is susceptible to mutation, then you can determine in absolute number of years the time that has elapsed since the taxa that you're comparing shared a common ancestor. Beautiful. So this is what's known as a strict molecular clock, where you have a single that is global rate of substitution for that protein and perhaps for other proteins as well. Zuckerkandl and Pauling thought that nucleotides were better than amino acids in this regard because any change in a nucleotide will be visible, whereas amino acids may mask, because there are synonymous codons for amino acids, may mask underlying changes and so hide them from you. So nucleotides would be better. So this was very popular and got a lot of attention. Of course, I'm sure you've heard of molecular clocks and thought about that this is basically the way that they work. But there's some significant issues here. First of all, when people started applying strict molecular clocks to problems that were thought to be fairly well known, such as the age of angiosperms, the age of mammals, and things like that, they found that there could be some pretty significant differences between the projected ages of these groups when they were based on fossils as opposed to when they were based on molecules. For example, for the age of angiosperms, the difference was between the two was like, what was it, 10, 15 million years. And so more than 10% of the total age that they differed. So that's a pretty significant difference. Secondly, in some comparisons, if the fossil calibration points, so the areas of the cladogram on which you had fossils that you could assign to that node and therefore link the age of the fossil to the age of that node. If they were out toward the end of the tree, closer to where your OTUs are, closer to the present, then there appeared to be a tendency to underestimate the age of the groups. And conversely, if the fossil calibration points were deep in the tree, far back in time, then there was a tendency to overestimate the age. We have to be careful about some circular reasoning here and where our evidences come from. So we have two competing non-ideal sources of information, basically, but they're not coming to the same answer. And then, of course, now you've seen this in your phylogenetic project. When you do an analysis and you have the branch lengths scaled such that they represent the amount of presumed evolution that has taken place along that branch, that a pair of branches that arise from a single node are not necessarily, in fact, are probably rather unusually the same length, which tells you that there is not a single rate of change that is applicable to all nodes across that tree, that there are different rates of change in different parts of the tree. And there may be different rates of change within a branch on a tree. You can imagine a protein evolving quickly early in the history of a species, and then for whatever reason, maybe it's reached a selective optimum. Evolution in that protein slows down, and we don't see any change. And so the idea that there is a single global substitution rate that we can apply blithely across the entire tree is clearly something that's not true and leads to errors that it would be nice if we could accommodate and control for that. This was just my example to show you here how this is that same tree you saw before, whereas here all the branches end at the same point. In these illustrations, the branch lengths are proportional to the amount of change. And you can see that we got some long branches up here, we got short branches. And look at that one right there, that we have a short and a longer branch coming off the same node. They're not evolving at the same rate. So the idea of a global substitution rate for a molecule, let alone across different molecules, is clearly untenable. So how do we accommodate heterogeneity and substitution rates? Well, first of all, why should heterogeneity exist? We don't have a definitive answer to this, but some suggestions have been made that have some empirical support and have their own criticisms. But typical suggestions have been four of them. One is that organisms with different generation times could differ in the rapidity with which the molecules, the proteins that make them up, can evolve. If you've got something that has a generation time of a week, then you have multiple generations. Differences can accumulate with mutations in them as they go along, because somatic mutations are irrelevant. It's mutations in the germline that are going to be passed on from one generation to the next and be represented in our trees. So shorter generation times could lead to faster substitution rates. Similarly, higher metabolic rates. I think this one has received a little criticism. One of the things that's true is that high metabolic rate seems to be correlated with generation time. So high metabolic rate, short generation time, and vice versa. So perhaps these are not two different things. The mutation rate themselves just inherently can vary. And then there's the idea of effective population size. So if you remember your population genetics, the probability that a mutation becomes fixed in a population is going to be a function of the effective population size. Higher probability of becoming fixed in small populations than in large populations. So effective population size is basically the number of reproducing individuals. So it's not counting post-senescent older individuals or pre-reproductive juveniles or things like that. So how do we accommodate the existence? We know that there is heterogeneity in substitution rates. How can we accommodate this? Well, one option is to basically chop up your tree. I'm going to stick with the tree metaphor. Chop up your tree into chunks in which the substitution rate within a chunk is more or less constant, or at least acceptably so. In other words, to go from a single global substitution rate to having multiple local rates, and then scaling the tree in that manner. Another option would be to prune and discard the branches of your tree in which you see the different rates. In other words, prune the tree, getting rid of the taxa that have these different substitution rates so that the only ones that are left all have basically the same substitution rate. This is a strategy called taxon shopping, which has been severely criticized because basically going and looking to find the data that support your a priori answer there, or at least you run the risk of doing that. A third option that I'm not going to get into the details here because it's way beyond the scope of this course, is that you incorporate these ideas of rate heterogeneity in your models, both maximum likelihood models, and now there's a plethora of Bayesian methods to do this. Basically, in the prior distributions that are the input into your Bayesian analysis, you incorporate this idea of heterogeneity and substitution rates. And then finally, one that was just recently been proposed is kind of the opposite side of the coin of taxon shopping, and that they called it gene shopping. Basically, now a phylogenetic analysis can include thousands of genes. Idea, you go in, look at each of those individually, and test them to see whether or not the rate of substitution is more or less constant throughout the evolutionary history of that one gene, or if it shows differences through time or across the taxa in there. And then you only take those and analyze those that have strict clock-like behavior in order to put absolute ages on your cladogram. Again, you're going to, in most of these, you're still going to need some fossil calibration points. Let's see what's next. So there are lots of techniques that do all of those things. There's one factor that I haven't yet talked about that I want to introduce here because we're going to talk about it a bit later when we talk about DNA barcoding. And that is a phenomenon known as incomplete lineage sorting. So when I heard that term, I had no idea what it meant by virtue of the words themselves. So I needed to have a graphical explanation of it. So here's my attempt to provide a graphical explanation to you of what is meant by the term incomplete lineage sorting. So the black lines here, well, let's start at the bottom. We have a simple 3 taxon statement for 3 taxa, 3 primates, the gorilla, chimpanzee, and human, for which I think it's well established that we understand to the point of actually knowing for sure what the phylogenetic relationships among these organisms are. That is, chimpanzees and humans are more closely related to each other than either is to the gorilla because they share a more recent common ancestor. This area up here on this 3 taxon statement. So the outer solid, oops, I lost my cursor. There we go. The outer solid black lines represent the cladogram, the 3 taxon statement showing the phylogenetic relationships among them. These inner circles and the lines connecting them represent for one gene what 4 different alleles for that one gene. So we start out, we have a single allele in the distant common ancestor of these 3 that then when, then this gene duplicates to produce this, what color is that? Pink, and this blue allele, and these are passed on to descendants. So we're going through time here, going down, we're going down, we're going forward in time to more recent times. Then we have another gene duplication event where this pink one gives rise to 2 daughter genes, 2 paralogs, one represented by black and one represented by pink. And over here, this blue one, gene duplication, or not gene duplication, we get a mutation that results in the origin of a new allele. So, excuse me, not gene duplication, mutation giving rise to new alleles. So here in the common ancestor, when we get down to this point, we have 4 alleles in this common ancestor. And then there's a speciation event in which the ancestral gorillas branch off, leaving the common ancestor of the chimpanzee plus humans on the other side of that split. And in this lineage that leads to the gorillas, only one of those alleles made it into what is now the modern gorilla population. Maybe this pink one did, but it was eliminated by extinction or just chance early on. And so in modern gorilla populations, we only have the black allele. In the common ancestor that eventually led to humans plus chimpanzees, we started out with 3 alleles, pink, yellow, and blue. In the chimpanzee, only the yellow made it to the modern populations. And in the human, although we started out with both yellow and blue, we eventually lost the yellow allele, ended up with only the blue allele. The pink allele got lost very early on here. So now we can see that the evolutionary history of this gene, an initial split that produces a black, and then another split, well, initial split that then led to the production of the black allele, and then this split which led to the yellow allele and the blue allele. The evolutionary history of this gene exactly mirrors the evolutionary history of the organisms at the bottom. An early split that led to the gorilla, and a subsequent split that led to the chimpanzee and the human. This is complete lineage sorting. And this kind of data, actually this kind of data down here, is what we hope we are using when we construct a phylogenetic tree based upon molecular data. But what about incomplete lineage sorting? Same scenario, except same scenario for the gorilla. But now over here, in this common ancestor of the human and the chimpanzee, it turns out that this pink allele was not lost from the entire population, but it actually, from an ancestor that had all 3, those that ended up in modern humans, only the pink allele made it all the way. We lost the blue along the way. The yellow never got in this lineage at all. And so we have the pink allele here. And then we have the yellow allele just as we did before. But you see now, because early on we had both blue and pink derived from this common ancestor that had all 3, now the evolutionary history of this gene contradicts or is not compatible with the relationships amongst the organisms. The gene tree does not reflect the species tree, is the jargon that's used. And when this happens, this can lead to confusion and difficulties in dating. So this is just showing you the same thing. If you want to pause this, and these 2 are examples of incomplete lineage sorting where the gene tree and species tree are not compatible. This is the complete lineage sorting where the gene tree and species tree are compatible. T here represents the time between the split from the common ancestor of ABC to, let me get this right. This represents the split from the common ancestor of B and C. To, and then tau BC is when B and C finally split from their common ancestor. I hope I explained that correctly. I'm sure you can understand it from what this tree. So this is the time between these two speciation events. What's the probability of coming up with complete lineage sorting? That's represented here, and I think you can see that if we reduce this time between these splits such that it disappears as t approaches 0, what is the probability of getting, or think about it this way, what is the probability of getting a tree that has complete lineage sorting? It's 1 minus 2/3 e to the 0, that would be 1, so that only 1/3 of your gene trees as t approaches 0 will reflect the species tree. Another way to look at that is 2/3 of the genes that you're looking at. As this gap in time becomes smaller and smaller, 2/3 will give you the wrong phylogenetic signal and also give you the wrong timing signal. This, it would be pushing the timing of the split back in time. So this leads to the conclusion, well, this leads to two things. First of all, that the problem of incomplete lineage sorting may be particularly important when we're talking about recent speciation events, recent evolutionary events. So if you're talking about within the last 1 to 10 million years, incomplete lineage sorting may be a severe problem. You go farther back in time, then these genes dominate the number of markers, the pattern of evolution of markers here. So we don't need to worry so much about incomplete lineage sorting at these levels. Second is that there are analytical techniques that look at this process of incomplete lineage sorting, or more specifically, to look at the way that these genes match up in common ancestors as you go back in time. This is called coalescent theory, and it's called specifically multi-species coalescent theory when we have more than one species involved here. And so multi-species coalescent theory has a lot to say about dating issues and inference of phylogeny when we're talking about these relatively recent events. So let's see, I said some of this already. I guess the main point I want to add down here is that there are still very, very few empirical examples to guide us about how to handle theoretical, or to what extent theoretical problems arising from our data and the way genes evolve impact these techniques we use in order to do things like trying to put absolute ages on speciation events. So we're still in a very early stage. I think it would probably be safe to say that no methods are particularly good at handling all of the issues involved with dating phylogenetic trees. They all have issues. You know, if you're going to use Bayesian methods, what priors do you put on? What prior probabilities do you assign to all the elements that go into this process? How do errors in the model specification impact the dates you come up with? All of these sorts of things. We're still in very early stages, and there are quite literally dozens and dozens of computer programs available that approach the problem from one aspect or another, but there's still very little I can say definitive. I guess what I will say is that when you look at a phylogenetic tree, at a chronogram, a phylogenetic tree that has time explicitly labeled on it, take it with a grain of salt, especially if those dates have no error bars associated with them. So, quick summary. How do we get from cladograms? How how to get from cladograms to classifications. There's a lot of room for disagreement. The only relatively firm criterion is that in a classification, all of the taxa that are formally recognized and named, they should be strictly monophyletic. But other best practices include minimizing the number of monobasic taxa that you include in a classification so that your classification actually does something. And then to be conservative, we have over a quarter of a millennium of history of the use of names, and there's a lot of value in that collective tradition of how names are used. And so be conservative. If you have to propose changes, keep them to a minimum and highly justify them. I didn't make this point earlier, but I mean, we do see reclassifications, and a high proportion of them, such as the Platygastroidea example I gave to you, come about because of the desire to eliminate paraphyletic groups from classification. Paraphyletic groups, in my mind, are actively misleading. They suggest that the species within that group all share something unique to themselves that nobody else has, which is not true, because the other descendants from that common ancestor that are excluded from the paraphyletic group also have that same evolutionary heritage. And then for dating methods, fossils, we know, give a minimum age for a taxon if you can confidently assign a fossil to a node in a tree. The initial attempts at using molecular clocks relied on strict molecular clocks, which have lots of problems. Now there are lots of other, they call relaxed molecular clock methods, both maximum likelihood and molecular methods, because, I'm sorry, maximum likelihood and Bayesian methods, because largely the data that go into these are based upon molecular sequences. But there's lots of issues, and we're still far from a consensus on the best way to approach this and understanding what the impact of errors are. For very recent divergences, then the methods associated with the multi-species coalescent theory are probably going to be pretty important for you. But for older things, such as the discussions we'll be having on the age of insect orders, then coalescence is not going to be, and incomplete lineage sorting is not going to be such a big issue. To close, I just wanted to show you a cladogram. This is a cladogram of an analysis of, I believe these were myrmecine. No, that looks like it's all ants, a wide variety of ants here. We can focus down here. There's lots of information being imparted here. Here's the absolute age in millions of years ago, so from 25, 50, 75 up to 125 million years ago, which is where they propose ants arose somewhere back here. But these horizontal lines here show you the error bars associated with it. And so in terms of the origin of ants, and they may be as old as nearly 125 million years old, or perhaps they're only, what is that, 75, 80, 85 to 90 million years old. So a range of 30, 40 million years, a pretty significant error bar, if you ask me. So that's the state of the art that we have it. So I'm going to sign off for now and look forward to seeing you on Thursday.